recording all of the sessions and then posting them. I just started recording now, so if you want to go back and look at this session again, you'll have the ability to do that. I'll send you a follow-up email where I'll describe exactly where these will be. My plan is to upload these videos to YouTube, and it'll be a YouTube link once I figure out how to do that. Okay, so I'll follow up after today's lecture and provide you information where these videos will actually be housed. Okay, so we're going to go ahead and get started here. Bear with me while I share my application. Okay, and here we go. All right, cool. So we're going to start talking about the muscular system. So I had sent in an email, a suggestion that you read in Chapter 6, the muscular system. I believe I said up to contraction of a skeletal muscle as a whole, which is on page 191. So hopefully you did that. And if you did do that, then you have at least been exposed to the material that we're going to cover today. Now, we will not be covering the entire muscular system chapter. We're only going up to muscle movements, roles, and names, which is on page 196. Everything after that is about gross anatomy of muscles and muscle movements, and that's what's being covered in your lab. All right, so there's no need to go beyond muscle movements, roles, and names. Actually, not beyond, up to. Don't go beyond that. Go up to muscle movements, roles, and names on page 196. Okay, so let's go ahead and get started. I always like to begin with the objectives. And we're going to talk first about the functions of muscles. We're going to describe the basic anatomy, functions, and locations of the 3 types of muscle tissue. And then we're going to describe the gross anatomy of a skeletal muscle organ. And they're going to compare and contrast a tendon and aponeurosis. So muscles are responsible for all types of body movement, right? The function of a muscle is movement. Now, the name muscle originates from Latin mus or mus, means little mouse, and it's because someone thought a muscle moving under the skin sort of resembled like a little mouse scurrying under the skin. As odd as that might sound, but that's where the name actually originates. Muscles cause movement by contracting or shortening. It is a distinguishing characteristic of muscle cells. No other cells in the body shorten or contract, and this is what enables movement. There are 3 types of muscles: skeletal, smooth, and cardiac. Skeletal and smooth muscle cells are relatively long and thin, and because of that anatomy, they're also referred to as muscle fibers. So when you hear the term or read the term muscle fiber, it is simply a skeletal muscle cell. Muscles contract due to the movement of myofilaments, also called microfilaments. If you remember back when we were learning about the basic anatomy of a cell, and we talked about the cytoskeleton, it had 3 components, and one of those were the microfilaments, right? The microfilaments enable cells which can move to move, right? And these are also called, or at least in muscle cells, they're also called myofilaments. So myofilaments, microfilaments are the same. Now, as we talk about muscles, you're going to find that a lot of the structures of muscles and of muscle cells have the prefixes myo, mice, and sarco. Myo and mice are just mean muscle, and sarco means flesh. The flesh of an animal is typically the part of the animal that you eat. And when you eat meat, for the most part, what you are eating are the skeletal muscles, right? Whether we're talking about chicken, or we're talking about steak, or pork tenderloin, what we typically eat is the flesh. It's the muscle. So myo, mice, sarco, all refer to muscles. Now, there are three basic types of skeletal muscle. There are three basic types of muscle tissue: smooth muscle, cardiac muscle, skeletal muscle. But I want to point out that only skeletal muscles comprise the muscular system. So when you hear the term muscular system, it only refers to skeletal muscle. It does not refer to cardiac muscle. It does not refer to smooth muscle. Now let's look at the three basic types of muscle tissue and talk about their characteristics. Smooth muscle, as you know, is called smooth muscle because under the microscope, it looks smooth. You don't have those striations that you see with skeletal and cardiac muscle. Smooth muscles cells tend to have one nucleus. As you know, they are involuntary. You cannot consciously control them. And they're found surrounding hollow organs like your entire gastrointestinal tract, your urinary tract, the pupil of your eyes. I tell students, if it moves, you can't control it, and it's not the heart, it's probably smooth muscle that's causing the movement. Cardiac muscle, like smooth muscle, is involuntary, but unlike smooth muscle, it has striations, and you saw that in lab under the microscope. Cardiac muscle is only found in the heart. It's found nowhere else, only in the heart. Also, a distinguishing characteristic of cardiac muscle tissue is there are intercalated discs between the skeletal muscle cells, the cardiac muscle cells. Okay. Also, some cardiac muscle cells are involuntary. I'm sorry. All cardiac muscle cells are involuntary. Some have what's called pacemaker activity, meaning they don't have to be stimulated to contract. They will contract on their own. Lastly are skeletal muscle. Skeletal muscle, like cardiac muscle, is striated. They are multinucleated, meaning they have multiple nuclei. Cardiac muscle cells have one or two. Smooth muscle only have one. And unlike cardiac muscle and unlike smooth muscle, they are under voluntary control. Okay, so smooth muscles you can't control, cardiac muscle you can't control, skeletal muscle cells we can control, of course. Okay, all right. Now, as I mentioned, only skeletal muscle tissue is part of the muscular system. So from here on out, we are specifically going to talk about the muscular system. So yes, we defined what is cardiac muscle, what is smooth muscle, and what is skeletal muscle tissue. But from here on out, we're going to focus exclusively on skeletal muscle and skeletal muscle tissue, because only skeletal muscle makes up the muscular system. Okay? All right, so let's talk about what is the functions of the muscular system. Whenever I talk about a organ system, I like to specifically talk about what are its functions first, because then everything else we talk about are going to be details that refer to their function. So first, the muscular system, of course, is responsible for movement, and we're specifically talking about like locomotion, right? Moving your limbs or moving your mouth. Maintaining posture. I know we don't think about this, but all day long, the muscles in the back of your neck, right, are allowing you to hold your head up. You may be sitting right now, but you're sitting up, and your ability to sit up is mainly due to muscles in your abdomen, in your back, that are contracting and allowing you to sit up. So the muscular system helps you maintain posture. Muscles cross joints, and not only do they move those joints, they also help stabilize those joints. So that's another function of the muscular system. And lastly, skeletal muscles make up about 40% of your body, and they up by weight, and they're always contracting. Even when you're sleeping, they are at least partially contracting. You always have a certain amount of muscle tone, that is muscle tension, and as they contract, they use ATP, and a percentage of that ATP, when it's used for contraction, is a percentage of the energy is released as heat. And so your muscular system generates heat to maintain your body temperature. That is why if you exercise, you generate heat and you sweat, because your body is trying to maintain normal body temperature. Okay, let's take a moment to look at a skeletal muscle. All right, let's make sure you understand what is a skeletal muscle. And this is skeletal muscle, the organ. So this is this image right here shows skeletal muscle, the organ, like your biceps brachii or your pectoralis major. So what actually is a skeletal muscle? Well, it's basically skeletal muscle fibers wrapped in various layers of connective tissue. And then that skeletal muscle, or the organ, is attached to a bone or sometimes cartilage by a tendon or a flat structure called an aponeurosis. And we'll talk about the difference between a tendon and an aponeurosis shortly. Let's look a little more deeply at the anatomy of a skeletal muscle. So a skeletal muscle, as I said, is basically composed of skeletal muscle fibers or cells wrapped in various layers of connective tissue. So you have an individual skeletal muscle fiber here, and then what surrounds each skeletal muscle fiber is a layer called an endomysium. So you can see the little endomysiums that are surrounding each of these skeletal muscle fibers rather. And then all of those skeletal muscle fibers are packaged together and surrounded by a layer of connective tissue called a perimysium. And this entire structure, the skeletal muscle fibers wrapped in the perimysium, is called a fascicle. Remember, fascicle means a bundle of sticks, so it's because it sort of looks like a bundle of sticks. And then you have several fascicles that are bundled together and surrounded by an overcoat of connective tissue called an epimysium. And that's actually what you see if you ever look at a skeletal muscle, right? You're actually looking at the epimysium. And then that epimysium is continuous with the tendon or aponeurosis that attaches it to the bone. Now, think for a second. What does this structure and the terms for these various connective tissue layers remind you of? Just think about it for a second. Answer to yourself. and then let's cover it. Well, if you said nerve, you're exactly correct. What is a nerve? It's neurons, or excuse me, it's axons packaged in various layers of connective tissue. And notice that, but for the prefixes to each of these names, the names are the same. In the nerve, we had a endoneurium, perineurium, and then epineurium. The only thing that we've changed is the suffix, all right? So if you have learned the structure of a nerve, it's not very difficult to learn the structure of a skeletal muscle. Okay, so I mentioned earlier that tendons and aponeuroses attach muscles to bones. Now, I think most of you are probably familiar with tendons, right? You can just look at your wrist and move your wrist, and if you have a little bony wrist like mine, right, you can see the tendons right up underneath the skin. Tendons are these cord-like fiber structures that attach muscle to bone. But in some parts of the body, rather than having ropey structures, we have flat structures. Like on our head, we have what are called aponeuroses. They're the same composition as tendons. These fiber structures with fiber structures with lots of collagen fibers for tensile strength. But instead of ropes, they're sheets. Right? You have aponeuroses that attach your six-pack or eight-pack, really, your rectus abdominis muscles to your sternum and your pubic bone. Right? So tendons are ropes, ropey structures. Aponeuroses are flat, but their functions are the same. They attach muscle to bone or other connective tissues. Okay? And so as I mentioned, that epimysium is continuous with a tendon or an aponeuroses, and they attach that muscle to bones. Sometimes they attach to cartilage. Muscles aren't always attached to bone and cartilage. Sometimes they're attached to other connective tissue coverings as well. Okay, so I want to take a moment here to answer any questions you have. So the situation is I'm not able to see questions when I am lecturing. So let me take a moment here to see what questions have been sent. Okay, so what questions do you have on the material we just covered? Right, raise your hand, let me recognize you, and then you can unmute your mic. No questions? All right, so this question is, is a tendon like a rope? Yes, it is. A tendon is like a rope, and the aponeuroses is like a sheet. But the functions are the same to attach the muscle to bone, cartilage, other connective tissues. Other questions? Okay, Don, you have a question? Okay, I'll address that question at the end. Anything that's not directly related to what we're talking about, I'll address at the end. We're going to have a 10-minute session at the end. No worries. Okay, well, we'll continue. Let's see here, share. All right, so we've talked about skeletal muscle, the organ. Now we're going to move and talk about skeletal muscle fibers, because that's actually where contraction actually occurs. So first we're going to describe the microscopic anatomy of a skeletal muscle fiber or cell. Then we're going to describe the microscopic anatomy of what's called a sarcomere. And then we're going to look at the thin and thick filaments of a sarcomere and how they interact to cause muscle contraction of the skeletal muscle fiber. And I should have mentioned this at the beginning of at the beginning of lecture, but you will need a plain sheet of paper for a little activity that we're going to do in a few minutes. Okay, so what you are looking at here is a skeletal muscle fiber that has been cut. So imagine we took a skeletal muscle fiber is thin and long, and let's say we went and cut it right here. So this is a nice cross section of the skeletal muscle fiber. A couple of things I want to point out. First, you've got lots of little nuclei, right? They're multinucleated cells. Also, notice that those nuclei are right under the sarcolemma or plasma membrane. The sarcolemma, plasma membrane, same thing. So usually the nuclei are in the middle of the cells, right? Usually that's where we find them. Why would they be just under the plasma membranes? Well, it's because if they were in the middle of the cell, they'd get in the way of contraction. Nuclei don't contract, they just get in the way. So they're pushed off to the side and are found right under the plasma membranes. Notice what fills most of the cytoplasm of the skeletal muscle fiber. They're these organelles called myofibrils. Here's one that's been pulled out. But notice that almost the entire volume of the cytoplasm is filled with these myofibrils. Obviously, these myofibrils are going to be important, and yes, they are. They are actually the organelles that contract and cause the skeletal muscle fiber overall to contract. Now, I want to call your attention to another important organelle found in the cytoplasm, and that's the Sarcoplasmic Reticulum. So the Sarcoplasmic Reticulum is nothing more than a specialized, smooth endoplasmic reticulum. How is it specialized? Well, it stores calcium, and you're going to see later today why that's important. If we were to look at these myofibrils and blow them up, you would see that surrounding each of those myofibrils, shown here in blue, is a sarcoplasmic reticulum. So we've got the cytoplasm filled with these myofibrils, and then surrounding each of those myofibrils, this sarcoplasmic reticulum, which stores calcium. Now, let's go back to our myofibrils, right? So here's a myofibril here shown on the right. Notice that you have some dark bands and some light bands. Dark bands and light bands. We call the dark bands A bands, and an easy way to remember that is A is the second letter in dark. And these light bands we refer to as I bands, and again, an easy way to remember that is I is the second letter in light. These dark and light bands are caused by the way the myofilaments arrange themselves in the cells. They arrange themselves in a way that you get that banding pattern, and that was what was responsible for the banding patterns, the striations that we saw in cardiac muscle tissue and skeletal muscle tissue in the tissues lab when we saw those tissues under the microscope. It's this banding pattern here. Now, notice running from, this myofibril is composed of subunits, which we call sarcomeres, right? So looking at this myofibril, if you look closely at it, you would see it's composed of these repeating units. Each of these units is referred to as a sarcomere. Okay. If a myofibril is a train, If a myofibril is a train, the sarcomeres would be like the boxcars. So a myofibril is composed of these repeating units called sarcomeres. Why do we care about sarcomeres? Well, sarcomeres is actually where contraction occurs. Each of those sarcomeres is going to contract or shorten. And as each of those sarcomeres contract, it's going to cause the entire myofibril to contract. And if all of these myofibrils contract, guess what? The skeletal muscle fiber is going to contract, and thus the whole skeletal muscle is going to contract. Okay? So to understand skeletal muscle contraction, we have to understand how the sarcomere contracts, because that's really where it's occurring. All right, so let's look closely at a sarcomere. And a sarcomere basically runs from I-band to I-band. So this is a sarcomere from here to here. And so then there'd be another sarcomere here, and there'd be another sarcomere here, and so forth and so on. Now, a sarcomere is where contraction actually occurs. So to understand contraction of a sarcomere, we have to have a basic understanding of the anatomy of a sarcomere. To understand the anatomy of a sarcomere, what I want to do is actually have you construct a sarcomere. So I want you to take out a blank sheet of paper. So I'm going to give you, hopefully everyone can do that in like about, I don't know, 30 seconds. Let me get you 30 seconds to grab a blank sheet of paper, right? It should be completely blank, right? Should be like that, okay? One of the best ways to learn the components of an object is to take it apart and put it back together, and then hope that you don't have any pieces left over. But even when you do, sometimes it still works, right? I can tell you that from personal experience. Don't know how it works, but it does. So we're going to do that. We're going to take the sarcomere apart, and then we're going to put it back together, right? So we've taken it apart. Here's a blank sheet starting with a blank sheet of paper, and then we're going to start constructing our sarcomere. You may want to use, it's not required, but you may want to use like a blue pen and a red pen. It's not required, but you might want to, but some students choose to do that. So give you about 30 seconds to get your blank sheet of paper, and then we'll continue. What I do? Trying to use my phone here to time, but okay, let's start constructing our sarcomere. So you're starting with a blank sheet of paper, nothing. So what I want you to do now is in the sort of the center of your paper, draw some thick lines. These are the thick filaments. Sometimes they're referred to as myosin filaments because they are composed of the protein myosin. In these slides, I'm going to use a red color because that's the color that your book uses to represent the myosin filaments. So just draw in a few of those. Doesn't have to be 5. It can be 2. It could be 3. It could be 4. Okay? But myosin filaments, or thick filaments. Now we're going to add the actin filaments. The actin filaments are the thin filaments, and as you can see in this on my slide, they're actually thinner. And notice they partially overlap the myosin filaments. Now the actin filaments or thin filaments are composed of the protein actin, and they're tied together here and here. And that location is referred to as the Z-disc. Now notice where we have the thick filaments, that is our dark A band, and where we have the thin filaments, that's the I band or the light band. So here's our A band, and here's our I band. And sort of like in the middle of our I bands, we have the Z discs. Notice that on the A band, we have an area here that in a uncontracted sarcomere, we don't have overlap between actin and myosin. And that's referred to as the H zone. Sometimes it's referred to as the bare zone. And what we know as a sarcomere runs from Z disc of one I band to Z disc of the next I band. Now, let me be clear. I am not going to ask you to label a sarcomere. I'm not going to ask you, well, what is a Z disc? What is the H zone or bare zone, right? It is important to understand the anatomy of the sarcomere in order to understand how a sarcomere contracts, which is what we're going to talk about next. Okay, in terms of, well, what do you have to know? You do have to know what an I band is and what an A band is. This other information I'm sharing with you, this exercise, is simply so that you understand the anatomy of a sarcomere, so that you can understand the physiology of contraction, how a sarcomere contracts. So now that you have a basic understanding of the anatomy of the sarcomere, let's move on and talk about the physiology, the contraction. So what you just constructed, this composes a myofibril, right? Each of these is a sarcomere. Boom, 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 boom. And as each sarcomere contracts, each myofibril contracts. So let's talk about the contraction of a sarcomere. Okay? Sarcomere runs from A band to A band. This figure comes from your book. Now we're sort of magnifying the sarcomere, right? So here's your myosin filaments in red. Here's your actin filaments in blue. Actin filaments are composed of the protein actin, as I mentioned. Myosin filaments are composed of the protein myosin. They also have enzymes which break down ATP to release energy, because as you know, muscle contraction requires energy. Now, something that I did not mention earlier, which I'm going to mention now, is the fact that where you have overlap in your sarcomere between the myosin filaments and the actin filaments, notice that there are these little extensions from the myosin filament called heads, and they extend toward the actin filaments. In fact, I'm going to go to the next slide because there's a larger image of this, right? So this is a sarcomere. We're still looking at a sarcomere. This is a sarcomere at rest, so it's not contracting. You know it's at rest because we have this bare zone here. Notice that where we have overlap between the myosin filament and the actin filament, we have these heads. Sometimes they're also referred to as cross bridges. Now, what happens during contraction? Well, what happens is these heads bind to the actin filament, and then they pull inward. They pull inward, and then they detach. On both sides, they're binding, pulling inward, and then detaching, right? So imagine this occurring across the surfaces of all of these overlapping myosin and actin filaments. So those heads are binding, doing what's called a power stroke, detaching, recocking, binding, doing the power stroke, detaching, almost like, I like it into like oars going in the water, and then you do your stroke, and then you raise your oar again, reposition it, and then drop it again. Well, what's going to happen as those myosin heads bind, do the power stroke, and detach? Well, what's going to happen is those actin filaments are going to move toward the middle, pulling the Z discs with them. And so you're going to get contraction of the entire sarcomere, right? So this is a sarcomere that's not contracted here at the top. This is a sarcomere that's contracted here at the bottom. Notice the Z discs are closer, and notice that where you had a bare zone where you have overlap, right? Now you have overlap. And so that sarcomere has contracted or shortened. And if all the sarcomeres contract or shorten, it's going to cause the myofibril to contract and shorten. Okay, this is referred to as the sliding filament theory of muscle contraction. Now I'm going to show you this video, which is going to review skeletal muscle anatomy and illustrate sarcomere contraction.
Skeletal muscles, like the biceps brachii.
Okay, bear with me just a second. I have to change the sharing here. Correct. The H zone does disappear. Let me show you this and I'll answer questions. Okay. Should pop up here in a second. There's a bit of a delay. Okay, why is it not sharing? Boy, well, it's a serious delay. All right, here we go.
Skeletal muscles, like the biceps brachii, attach to bone via connective tissue called tendons. Muscles are composed of bundles of muscle fibers. Each bundle is separated by connective tissues known as perimysium. Each fasciculus is made up of muscle fibers, which are separated by connective tissue called endomysium. Skeletal muscle fibers or cells are multinucleated and striated in appearance. Muscle cells are composed of subunits called myofibrils. Each myofibril is made up of several myofilaments. The two types of myofilament shown in red are composed primarily of the protein myosin, and a thin myofilament shown in blue composed mainly of the protein actin. The repeating arrangement of thick and thin myofilament serves as the fundamental subunit of striated muscle contraction. These subunits are called sarcomeres. A sarcomere contraction is represented by the shortening of the distance between the Z lines. The sarcomere shortens because the thin filaments slide past the thick filaments. In 3D, each thick myofilament is surrounded by 6 thin myofilaments arranged in a hexagonal pattern. The 3D arrangement of sliding myofilaments is the microscopic basis of muscle contraction.
Okay, so
sliding myofilaments is the microscopic.
All right, so I can pull this back a little more. All right, maybe a little more. All right, so it's these thin filaments being pulled inward that is the basis of muscle contraction, sarcomere contraction. As the sarcomeres contract, the myofibrils contract, and the microfilaments contract. I'm sorry, the skeletal muscle fibers contract. Now, I did a question about where can you find these videos? I'm glad you asked that question. If you go on Blackboard, there's quite a bit of delay between when I share something when it actually pops up. But anyway, and you go to the muscular system, all of the videos that I'm going to share with you in my presentation are going to be found here, right? So here are the slides, and then here's the video I just saw, and I just showed you, and these here are the videos I have yet to show you. Okay, so all of the ones I'm sharing with you, you can pull up as well on Blackboard. Yes. Okay, let's see where we are here. Okay, so we only have 2 minutes left, so I can't really start the next topic. Let's sort of review what we talked about. So we discussed first we discussed the 3 different types of skeletal muscle tissue. So make sure that you're familiar with the 3 different types and how they compare and contrast. Smooth, cardiac, skeletal muscle. Then we looked at the anatomy of skeletal muscle, the organ. Then we focused on the microscopic anatomy of a skeletal muscle fiber. We talked about the myofibrils, the sarcoplasmic reticulum, and although the focus of lecture is not anatomy, it's important to understand the anatomy to understand the physiology. We talked about the myofibrils, that they're composed of sarcomeres. We looked at the anatomy of the sarcomere. And then finally, what this was all about was understanding how these myosin heads interacted with the actin filaments to cause contraction. Now, we talked about how contraction occurs, right? The sarcomeres contract, causing the myofibrils to contract, and if all those myofibrils contract, the skeletal muscle fiber as a whole contracts. What we haven't talked about is, well, what causes those myosin heads to all of a sudden decide to bind to the actin filaments? And that's what we're going to talk about on Wednesday when we talk about motor units and how a motor neuron stimulates this process to occur in a skeletal muscle fiber. So please go ahead and read, and I'd say go ahead and read the remainder of the chapter that you're responsible for reading up to muscle movements, roles, and names on page 196. So read up to that. Don't read beyond that. Okay? So the formal instruction component of class is ended. So for the next 10 minutes, reserve the time for answering questions. And I see there's been a couple. All right, I talked about the link to the video. All right, so I have a question from Michelle. You can chat or you can mute your mic and ask your question. Michelle?
Hello?
Yes.
Okay. I was just wondering because you had said that it was an H zone, and in one of the slides it says inline.
The H zone, the air zone, it inline, right? It doesn't really matter what you call it, because I'm not going to ask you about it. It's just understanding that it's, you have that area of where there's not overlap when the sarcomere is not contracting, and then it's missing when after contraction. And right, as long as you see that, that's really all you have to know or realize, more or less. Okay, so there's a question about exam exam 2 makeup. So there are some students who never took the second exam, right? And so in the email I sent you this morning, I mentioned that that it was only for students who haven't taken exam 2. So those of you who have taken exam 2, one, you're not going to be able to retake it because it's password protected. Right? So don't worry about exam 2 that's posted under assignments if you've already taken it. That's only for the students who, no worries. It's only for students who haven't taken it. Now, I did post the exam 2 practice exam, and it was really for students who have yet to take exam 2. But if you want to sort of see what's going to be the setup of exam 3 online, it's going to be the same as the practice exam 2. So that might be something that you might want to check out. Take-home exam. The question, when is the take-home exam due? My thinking was to keep it for the same same deadline. I don't have a problem necessarily with extending the deadline. I just figured that, you know, probably you have more time, not less, with you know, the sheltering in place or whatever it's called now. So I was going to make it the same deadline. What I am going to do, however, is you're going to submit this online. So I'll actually be placing the take-home exam online, and then you can go and put your answers in. I got to talk. I'll talk a little bit more of that when it's ready to go, because I'm going to set it up so you can go in, submit some of your answers, save, and then come out, and then you can go back in and out at will. And you only want to submit it when you're completely done, because once you submit it, if there's anything you want to change, you're gonna have to redo it all over again, right? I do not remember when your take-home exam is due. I would just say look on it because it's on the first page. It's the date of the week before the original date of your last practical, whatever that date is. Question from Rachel. Let me talk to you. If you have some time in a few minutes, let's talk, you and I, specifically about that. Okay, Natalie thinks it's due April 29th. Let's see here. So I have April 29th and May 1st, depending on when your lab was. Of course, now I'd probably like make it May 1st because you're no longer going to lab. As we get closer to that date, if we need to push it back, I mean, I don't have an issue with that. Right now, we'll say May 1st, and if we need to push it back further, I'm just fine with that because it's going to be all on Blackboard. It's a click of a button to grade it. Okay, I'm going to go ahead. I'm going to stop the recording.